Okay, everyone, it's 8 o'clock. Maybe we'll give people one more minute to join in. In the meantime, I can just run you through a little bit of the logistics and just a general introduction. Today we're going to be talking about the phylogenetics of Coronaviridae. It's our fourth session on the coronavirus pathogen, and we're going to be introducing today the different phylogenetic tools and tree viewing tools that are available on the ViPR website. My name is Ananya Widomska. I'm the outreach coordinator for the virus pathogen resource, and joining me today is Richard Sherman, the head of informatics at JCVI. And our presenter today will be Christian, who's a senior bioinformatics engineer at JCVI and an expert on phylogenetics. So just a little bit of logistics will be, if you'd like to ask a question, I'd like to remind you to please ask it in the Q&A box, because it's a little bit hard for us to see it otherwise. Or if you'd prefer to ask your question during the Q&A time directly, you can just click on the button to raise your hand and we can unmute you, and you are more than welcome to just ask your question orally. The structure of this presentation will just be a brief introduction and walkthrough, followed by any short Q&A. And then Christian will walk you through a short tutorial of how to use the tree building tools on the ViPR website. And this webinar is, of course, being recorded, so it'll be available on YouTube shortly after the webinar for those of you who would like to watch it again, or for any of your colleagues who couldn't make it. And just as a quick reminder, we do have a couple other Coronaviridae scheduled workshops. November 3rd, we will discuss host response omics data in relation to coronaviruses. And on Tuesday, December 1st, we'll be discussing Coronaviridae ortholog group prediction. So if you haven't already registered for those and you're interested in what ViPR has available for those, you can go to the ViPR website or follow our social media streams to just keep up to date on those. And with that, I think we can go ahead and get started. Christian?
The topic is the phylogenetics of the Coronaviridae, which obviously currently is a hot topic. So, but it's not only, I mean, it's basically how to use phylogenetic tools in ViPR, so it applies also to other viruses, not just Coronaviridae, but I'll be focusing on the Coronaviridae. So, my name is Christian Smolzek, as already mentioned. So, just go over, so the presentation is, first I very quickly go over a couple of the theory, the basics. For probably most of you, it's a reminder what you already know, but maybe some people are new to this topic. So for those, it's a very quick introduction. And also, I will also later, depending on the question I'm getting, I will also produce, or I already produced a handout, but depending on what kind of question I have, I will add more to it, and this will be emailed to you maybe today or tomorrow. So, you get all the URLs and the data on a PDF. So, you don't have to take notes now or anything like that. So it will be emailed to you, but depending on kind of what the question, I might add something to it. So, okay, so the basics. So the phylogenetic trees are diagrams which show the evolutionary relationship of species or genes and proteins. And if your tree is made of species, it's called a species tree, or it can be a gene tree. So those are and for any given family or species, they don't have to be the same. So the species tree can look different than the gene trees due to gene duplication or gene loss. And a very important point is that phylogenetic trees can only be inferred from homologous sequences. So for example, in case of the Coronaviridae, for example, you can build a tree based on the spike protein, the S proteins, but you cannot mix different proteins together and build a tree. So there has to be only one. Or you can also build trees, if the genomes are very similar, you can also build trees on whole genomes. And the basic approach for tree building is, first you have to obtain the sequences to be analyzed. And as I said, it can be either individual genes or it can be whole genomes. And this step might depend on your study, might be actually quite time consuming and difficult, because the family you want to study, there might be thousands or tens of thousands of proteins, but for technical reasons, you might not be able to analyze all of them. So you have to select a representative subset. And then the second step, of course, is you have to create a multiple sequence alignment. And then as a final step, you have to calculate the actual tree. And just an example of some of the programs which you can use for MSA calculation is MOFT, MOSSEL, or CLOSSEL Omega. And I underlined those which are available in ViPR. So in ViPR, MOSSEL is available. Currently, MOFT or CLOSSEL Omega is not available, but at some point there might be. And same thing for tree building. I mean, there's a greater variety of methods. On one hand, you can build trees based on pairwise distances, which means you have to calculate the pairwise distances first. And here the most well-known method is neighbor joining, which has been implemented in ViPR in the form of quick tree. And then the more accurate methods are the character-based ones, maximum likelihood, maximum parsimony, and Bayesian methods. And here ViPR offers RexML and PhyML as examples of maximum likelihood-based methods. Once you have to calculate the tree, you have to actually analyze the tree, and that's obviously, it's manual work. And here many people make mistakes. And I would like you to point to this very important paper or summary, which is called Understanding Evolutionary Trees, which tells you, about common mistakes people make when they look at phylogenetic trees. And this paper has many examples of errors. When you're looking at a tree, what kind of errors people make. And I'll give you a couple of examples, but there's many more errors you can make. And please read this paper. It's easy to read, and it really is very useful. So basically, how to read evolutionary trees. So just quick reminder of the basic terms. So usually a tree, in most cases, the trees are rooted. So they have a root, you have branches, you have terminal nodes, which sometimes also called tips. You have internal nodes, and oftentimes you have an outgroup, which is a species or a gene which is more distantly related to the rest. And this is used for rooting the tree. So that's that. And then you have, sometimes people call about sister taxa when you have a relationship and taxa are related by, you have a parent, and you have A and B here, so those are called sisters. So that's that. And then another important term is clades. So clades can be hierarchical. So for example, here we have a clade corresponds to this subtree, A, C, and K. And another clade is P, Y, and S. And then the whole tree is also a clade. So as I said, the clades are hierarchical. You have like higher level and lower level. And more important in a way is actually how to not read evolutionary trees. And here I just give a couple of examples. As I said in the paper, there's many more. So one problem is people are reading across the tips, which means is usually oftentimes a tree is drawn like this. And then people might wonder, is the frog more closely related to a human or to a fish? And if you just look at this naively, you say, a frog is most closely related to a fish because they're just neighbors on this, the way it's drawn. But actually to determine the distance, you have to follow this path. So from the frog, you have to measure this one, you have to measure this one, and this one. So that gives you the distance to the human, and then the distance to the fish is like this one, this one, and this one. And you can see this distance is actually longer. So frog, of course, is more closely related to humans than it is to fish. And you can draw the tree differently, this exact same tree. And you can see now this, in a way it's more obvious. So here you have frog and human together. So that's a very common error, and it's very easy to make actually if you're looking at very big trees. When the internal structure, because you have so many, branches, the whole tree is like really, you can almost not see the branches anymore. And then it's very easy to make this mistake. And so that's a common mistake. And then another thing is like the amount of evolution a certain gene or an organism underwent. And here again, if we look at human and compare with a fish, you think, oh, the fish it underwent less evolution because there's no branching on, or there's no branching on this branch leading to the fish. Whereas the human has many, it looks like it's more, it's more evolved or has more, underwent many more steps of evolution. But then if you can draw different organisms. So now you're interested in echinoderms, which are sea stars and the sea urchins and sea cucumbers, and then now it looks suddenly human is more, it underwent less evolution. Again, it's of course not true about it. So it's just a way of drawing the trees, and so those are just two common examples of errors people sometimes make. And now I'm going to the live demo. So what I'm going to do is I very quickly remind you about how to use ViPR. And since the actually, selecting the sequences, what you want to analyze is relatively time consuming. So my first example, I will start where I already collected a set of sequences and then just show you how to build a tree from it. And then as a second example will be, I show you how to select a couple of, sequences and then building a simple tree. And as finally, at the end, you can calculate your own trees, and I'll be available for if you have any questions. So now we go. So that's of course the URL for ViPR. So we should go. I will go there now. So now you should see basically. So this is what you should. I hope everybody. You can see a Coronaviridae from ViPR. So just show you how you get in there. So this is the ViPR website, and I strongly and already covered this in previous workshops, how to use search and also how to use the workbench. So basically how you search is you select the virus family, and then you can go here, or if you haven't selected, you have to, it forces you to select it. And then here you can, for example, you can search for proteins, and then here have the different subfamily of the Coronaviridae. You can open this one here, and then you could collect certain sequences you want to analyze. But as I said, I'm not going to do that because it takes a while. The more important thing is, as I already mentioned, you can only build trees on homologous sequences. So that's, for example, here I use the spike protein in that example I'm going to show you. So that's, how you would do that. So as I said, I already made this. So I already selected certain sequences. Here is 24 proteins which are selected, and they are, they cover a couple of different species from Coronaviridae. And then what you would do, so you have your proteins, then you select all of them. So now you see there's a check mark here, so that means they're all selected. And now you can analyze them. And then for this one, you go to this one here, the run analysis box, and you can either directly build a phylogenetic tree, which automatically also creates the multiple sequence alignment, or you can first create the alignment. So I'm showing you how you do that. So I'm first building the alignment, because it's better to look at the alignment and make sure it's good. So you click here, and then you can choose whether you want to build the alignment based on amino acids or CDS. So here I select amino acids, and then you go continue. And then here it doesn't actually give you too many options. So you can give a name to the analysis, and you can select which field actually, in the labels what you want to display. For example, if you want to have the date there or the country. But I just leave it as it is, and then that's pretty much it. And then you will click on run, and then it would it will calculate the multiple sequence alignment, which again is probably taking a while. so I'm going to show you. Oh no, it was pretty quick. So it is done. And then now here we can see the multiple sequence alignment in this tool, in this MSA viewer tool. And with this one here, you can kind of scroll through it. You can look at it. And here is a good point is to actually inspect the alignment if it's good or bad. So if you see a lot of gaps, I mean, not necessarily meaning like a nice block of gaps, but if you see kind of random gaps everywhere, and there's almost no columns which are, which looks aligned, then maybe you have to kind of rethink your strategy and maybe have sequences in there which are too distantly related that they could not be aligned properly. But to me, this alignment looks pretty good. It's relatively long, but you see here you have nice blocks of good alignment where the alignment seems pretty decent. And so I think it can be used for tree building. And here you see here you have the labels. And yeah, so then the next step is to actually calculate the tree. We can also save it if you want, which is probably a good idea. It goes to your workbench, but I already did that, so I'm not going to do it. So basically then here you go generate phylogenetic tree. And now here's a lot of options. And as I mentioned, there are simple methods for tree building like neighbor joining, or more advanced methods like maximum likelihood-based methods. So then here, so if you're not sure what you want to do, you can always do first, you can do quick tree, which is very fast, and you get your tree. But if you're serious about analysis, I strongly recommend you to do to use the custom tree method. And then here you can select if you want to use PhyML or XML. And if you want to do bootstrap analysis or not, and I always recommend to use bootstrap analysis because that shows you, how reliable the tree is. And then for maximum likelihood, you also have to choose the evolutionary model. I usually, for most viruses, I just use the LG. But if you're not sure what to use, you can also use protest to determine which is the best model. But I have to warn you, that takes a long time. So, but in general, I just use this one. And for me, I don't really care too much at the beginning because at the end, what I'm looking at is the bootstrap support. And if the bootstrap support is good, I trust the tree. And if it's not good, then I have to worry something could be wrong. So for example, the alignment could be bad, or maybe I used the wrong model, or there are sequences in there which are, don't belong in there because they're too distant. So, but I think it's not worth worrying too much at the beginning because as long as you calculate support values, it will tell you what you did is reliable or not. So as I said, I will not, at this point, I would just calculate initial tree and see how well supported it is. And okay, and so now we have done this. So we select the custom tree. We just use the strain name as a label. We use our XML. We perform bootstrap. We do 100, and then we build a tree. And now this one, even for this small tree, it actually takes quite a while. So I would say at least 20 minutes. So obviously we're not going to wait 20 minutes. So I'm already going to show you the finished tree, which actually looks like this. So and now I'm going to show you just how to use this visualization tool, which is called Archaeopteryx.js. And this actually has quite a bit of functions. So one thing you should always do is the tree, when it comes out of a program, it's always randomly rooted. So you can always reroute it. And one method to reroute is it's called a midpoint routing, which places the root in the middle of the tree, which is usually a good way to do if you don't have any better idea of routing. So for midpoint routing, you can click here, M, which stands for midpoint reroute. And now so the root now is placed in the middle of the tree. And now I'm just, I quickly go over all the buttons here, what they're doing. So this this button on top, they tell you what kind of diagram type to use. So this is it's called a phylogram. So here the real branch lengths are used for the tree, but you can also use a cladogram, which doesn't use the real branch lengths. It just shows you the topology, but the real branch lengths are ignored. And you can also do an aligned phylogram, which just means it's the same thing as the regular phylogram. It's just the labels are aligned. But usually, if we have branch lengths, I just use the phylogram. And then these buttons here, they control what you can see or not. So, for example, the node name, you can, if for some reason you don't want it, you can turn it off. So sometimes you have the sequence succession number. With this one, you can turn this one on and off. And then since this has confidence value, the bootstrap values, if you don't want to see them, you can turn them on and off with this button. You can also show the branch lengths or not show them. And then also, if you want, you can, draw the external node as little nodes or not. And same thing for the internal node or not. And this one here is, I'll show you this one later, node visualizations and branch visualization. And this dyna height, it's called for dynamic height. That is used if the tree is very big. Because if the tree is very big, if there's just too many labels, it will only show a few of them, and this you can turn this one off. But this tree is not very big, so you don't see any, you cannot, it doesn't have any effect. And then some of the viruses have really long names. So if you want to shorten them, you can do that. Then it just shows the beginning and the end of the name. So because it's like, like for example, this one is really long, and sometimes you don't want to show, not really care so much for the name, so you can shorten it like that. And then with this one here, you can zoom in different directions. You can zoom in this direction, and you can zoom in this direction. And with F, it's for fitting. You can always fit the tree, so it just uses the size. So with this one, you can control. And then these here, as I said, these are the tools. So one tool is a midpoint routing, and this one is you can order the branches so it looks a little bit nicer optically. What you can also do with these sliders here is you control the size of various things. So we show branch lengths. You can individually control that one. So those are pretty simple functions. One very useful function is the search function. So you can, for example, you type in, a name or something, and it will search for it and it will highlight it. And you can do two different ones. So like, let's say, so you can, you can highlight two different groups if you want to compare them or you cannot find them. This is very useful. And if these are, you can remove it. And then you can also, these buttons here control how you do the search. So for example, if it's case sensitive or not, if you only search for whole words, you can also, invert the search. And you can also use regular expressions. And you can also, if you want, you can collapse the tree. So you can have, you collapse subtrees. And you can also collapse by feature, by, for example, by year or country. And finally, you can also download the tree. And then you can also click on nodes. And, you can do another, you can do certain operations. For example, you can go to a subtree. So you only go, you show this subtree. And then if you want to return, you can click here, your return. And so now I show you one relatively unique feature of ViPR and Archaeopteryx is actually, so what you can do is you can visualize certain properties. For example, you can visualize according to the host. So you go here and you say, and then you say the label color is the host. And now you can see the hosts are, that use different colors. So for example, bat is red, or camel, for like a mouse, is similar color, so it's kind of unfortunate. And so if we click on this one, we can change the color. So for example, we can make this one, let's make it green. So this one becomes green. Chicken, common moorhen, let's make it another color. So you can change the colors like that of the for the host. You can also visualize other things, for example, the country where the virus has been isolated, for example, Brazil, China, Hong Kong, so on. And again, you can click on the nodes to change the colors. And also you can, the years, when it has been isolated. So depending on what kind of data is available, you can visualize it. And if you want to visualize multiple properties, you can, for example, use one of them to, the label, and then another property, maybe you can use, for example, the node itself. So, this one, then we can label according to, well, that's stupid, like make this the host. So you can do that. So you can visualize multiple properties. And with this one, you can move the labels around. And you can also visualize amino acids. Turn this one off. So this one, it kind of goes, it's sliding through the multiple sequence alignment, and it shows you the amino acid at any given position. So you see certain positions have a lot of different amino acids, or certain other positions only very limited ones. So that's pretty much it. And as I said, I'm actually the author of this program. So if you have any questions, you can email me, and I'm happy to help. So it has quite a bit of functions, but it should be relatively intuitive to use. And of course, you have to play around with it to really learn how to use it. So now I'm actually going to, this G I've been showing you, I have actually a little bit nicer picture where, which I colorized according to, alpha, beta, and gamma Coronaviridae, and I'm just going to show you this. Switching back to PowerPoint now.
Hey, Christian, there's been a series of questions in the Q&A box about what do the branch lengths correspond to in these phylogenetic trees?
They correspond to the distance between sequences. So the more distance two sequences are, the longer is the branch. The branches are. So if you have sequences which are almost identical, the branch will be very short. And if they're very, very different from each other, they will be long. And since sequence change happens over time, it also corresponds to, in a way, it indirectly also corresponds to time. So if usually if a branch is longer, that means, the time between parent and child is longer than a branch which is shorter. So primarily they simply correspond to sequence dissimilarity, but also to time. In the case of a phylogram, of course, if you have a cladogram, then they don't mean anything. They just show the relatedness, but the length is meaningless.
And when you say distance, depending on the method that's used to calculate relatedness, those distance measures can be different. Is that correct?
Yes, that is correct. Of course, in this short presentation, I don't have time to get into it, but there's many different ways of measuring the distance of sequence similarity. So the very simple models, you would just count how many positions are the same and how many positions are dissimilar, and then just calculate the ratio. But that's obviously it's too simple to be accurate. But that's a very simple way to do it. And then people use many more advanced maximum likelihood models to actually calculate those distances. But it is true. It really depends on what method you use to calculate it.
Mm-hmm.
Okay.
Just another quick question before you move on. There's another question that says, what is the significance of the horizontal lines between two strains or species as compared to the vertical lines.
Okay, let me switch back to, it's better to show with an example. So on this example here, so this, I mean, in this case, it depends on which direction you draw the tree, but so here the horizontal lines, so those are, they are, that's what we just discussed, they, correspond to sequence similarity. So the shorter that branch is, the more similar two sequences are. For example, if we compare this sequence here, so we will measure this part here and this part here. But then the vertical lines, I mean, they're just used to draw the tree. They don't really, they don't correspond to the, I mean, if we measure the distance, we would only measure this part here and this part here. We ignore this one because it's just used for drawing of the trees. So, or for example, if we are interested in comparing, let's say this sequence here, if this sequence here, so we will measure this part here, 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 and this part here. And so the vertical lines are just for the purpose of drawing. So they don't have any biological meaning, so to speak. So it's only, I mean, those are only the ones which, you see the branch lengths here. Those are the ones which are real. So, or again, comparing this one, we measure this one here, measure this one here.
And then there's a follow-up question that says, what is the best practice for selection of the bootstrap value?
You mean how many bootstraps you want to calculate, or what's the threshold where they become reliable? I mean, I can answer both questions. So in general, you should calculate at least 100 bootstraps. So most people in publications, they either do 100 or 1,000. And I don't think it really matters. I don't think they become more accurate if you do 1,000 instead of 100. Only thing it means your calculation takes 10 times longer. But you shouldn't do less than 100, and you should not do weird numbers like 55 or something like that. So it's standard to use either 100 or 1,000. So, but according to what kind of, what does it mean really? That's actually a very good question, and actually nobody really knows, because the bootstrap value don't really correspond to probability. So it kind of depends on, most people would probably say something above 90 if you do 100, let's say 90% is very reliable. And usually most people would say below 50% is not reliable for any given branch. But you see here we have mostly 100, so that means that would be very reliable. I would say anything above 90 is very reliable, and anything below 50 is not reliable. But then the area between 50 and 90, to be honest, I don't know, and I don't think anybody really knows. And it probably mostly depends on if you're publishing something, it depends on the reviewer what they think is good enough. So, but I mean, if you really have an important new finding, and you say, oh, I figured out this, an unusual relationship nobody expected, then your bootstrap values better be almost 100. So if you really if something really new you want to show. So here you see we have one relatively suspicious 168 here, but all the others are pretty good. But for example, this one here 71. I'm not sure if that's, I could not say, to be honest, is it trustworthy or not? I don't know.
And then there's a follow-up of, how about if I do 10,000 bootstrap value? Does that mean the tree would be more accurate as compared to 1,000 values? Or is there like an upper threshold?
No, if you do more bootstrap, it doesn't make the tree more accurate. It just means the bootstrap values themselves might be more accurate. It doesn't help you. Yeah, I know it sounds kind of like, oh, let's do more than the tree will become accurate. But that's not true. So, It doesn't make the tree more accurate. It's just, you can say that the bootstrap values themselves are more reliable.
So for coronaviruses, would one way to kind of make the relationship more accurate be to do trees based on multiple proteins in the virus?
Yes, that would be one way. If for example, what you can do is you can either concatenate the proteins, and then you make artificially longer sequences. That would help should help if the support values, or make it more reliable. Or you can either, personally, I wouldn't do it. I would do it differently. I would calculate a tree for each protein individually, and then compare those trees and see, where do they correspond or not. I'm not a huge fan of concatenating sequences or whole genome alignments. So I personally would then, it's more work, of course, but I think it's better because then I also get interesting information, because some proteins might actually be different from the rest. So then that in itself is biologically interesting. So that's what I would do. And generally, longer sequences give you a stronger signal. So if you have different proteins and you can only use one, and generally you probably use the longer one if you're trying to infer a species tree.
And then there was a question about, given the large number of methodologies for tree estimation, are there guidelines on how to know which method would be the most appropriate for a given task or data set?
That's again is a good question. I would say it depends on the size. The problem is it depends on the size of your data set. If your data set is huge, some of the more accurate methods are simply not available because they're more time consuming. But if you can, I would say something a maximum likelihood method like RAXML is probably one of the best you can do nowadays. And also RAXML is really fast. Another really great program is Mr. Bayes, which calculates Bayesian trees, but that unfortunately is not part of ViPR yet, but it might be in the future. But that's another really great program. But if you have a really big data set, or you have not much time, then you can use neighbor joining, which is not too bad. So I think.
Yeah, I think neighbor joining is usually referred to as the quick and dirty methods, right?
Yes, but the thing is, it's not really. That's what I tried to say before. It's not really worth, before you're building your trees, it's not worth really wasting too much time thinking about what method are you going to use, because the important things are the support values. So I always just use a method and calculate the tree and see what are the support values. And then if the support values are poor, then you have to think about it. Why are they poor? So the first candidate is the multiple sequence alignment might be bad. There might be wrong sequences in there. But it might also be that you need a more accurate method. For example, as I said, if you just use neighbor joining, maybe try something like RAXML. But there's huge papers published and huge arguments fought about which method is best. But I think if I had time, I probably would not focus so much on the tree building method. I'm probably more focused on getting the alignment correct, because that's in a way the kind of the important step. If your alignment is good, if you have only sequences in there which should be there, then it should not matter too much, as long as you use any kind of modern accepted method. So, and we hope that actually in the future that ViPR will have, it will give you more choice in both in multiple sequence alignment and also in tree building methods. So you have more choice, and especially I hope maybe one day there will be Mr. Bayes.
And just to clarify, ViPR has which methodologies available right now?
So currently it has for multiple sequence alignment, it has one, which is Mossel, and for tree building it has Quick Tree, which is an implementation of neighbor joining, and it has two maximum likelihood methods, RAXML and PhyML. So it has a total of 3 different tree building methods. And if you want to publish anything, I strongly recommend you to use to use a XML with 100 bootstraps. I'll just show it because time is kind of getting close. So I'll show you. As I said, I just quickly wanted to show you how you could build a tree, from scratch, because I didn't really show that. So what you would do is, and this is the issue of selecting the sequences, which is in the case of the coronavirus is difficult, especially if you're interested in SARS-CoV-2, because there are so many sequences in there now. So the important thing is, if you do protein, if you work on proteins, you have to, as I said, you can only align the same protein. So for example, we use the spike protein. So here, you can type in the gene symbol if you want to analyze. So using the spike protein S. And then here, you see, here you can open this box, and then you see the 4 different alpha, beta, gamma, and delta, and and that's the genus. And then you can also open this one here, and then you have the different species. Unfortunately, it doesn't really have a concept of strains. So that's, again, as I said, that's the problem with SARS-CoV-2. So what you can do is, what you can do is, so we select all the beta coronaviruses, and then you can, for example, if you know the name of the strain, you can type it in. 2019 new coronavirus. So how they show up here. So that's obviously the original Wuhan strain 1 and 2. So you could select this one here. Okay, so as I said, that's why I pre-made a tree, because as I said, it's not selecting your sequence is time consuming. It really depends on your question. So, but let's say we want to study the human coronaviruses 229E. We could select them all here, and then you do search, and then you see now you see the sequences that's just for 229E. And then what you could do is you select the ones you want, or if you're not sure what you want to do, you maybe just select a couple of representative ones. You select them here, and then you would add them to a working set. You maybe whatever you call it, 1. So then you add this to this working set, and you can go back and you search other sequences and add them to the same working set. And at the end, you can you go to your workbench, and you see this set here. You look at it, and there should be 4 sequences in there. That's obviously too simple an example, but that's how you do it. You build your working set from different groups, and then you could, again, as before, you say select all, and then you can, for example, you can go directly if you don't want to bother with looking at the multiple sequence around, which I don't recommend. But if you're in a hurry, you can just go here for generate phylogenetic tree, select amino acids, and then you would do like quick tree or custom tree. And again, I would do XML, bootstrap support, and then you're building your tree. So I think that's about it. And if you have any more questions, please ask them. And as I said, I will provide today or tomorrow. You will receive a handout which has some of the questions in there, and also links to maybe the paper I said, and a little bit more information about how to do it. So, and again, you can also
another attendee would like to ask a question.
Okay, so I am a bioinformatician at National Institute of Health, NIH, Islamabad, Pakistan, at virology department, and we are sequencing SARS-CoV-2 via ISEC 100 and MySeq. So my question is, previously, 2 weeks ago, I used ViPR, where you actually, the special one inside the ViPR, where you browse the annotation, assembly, and SNP analysis, which actually takes you to the Patrick browser as well. So my question is, when you upload your sequences, the forward and the reverse sequences in FASTQ format, so what is the policy of ViPR? Does ViPR actually store these data inside its database, or they just delete it? So I mean, I'm the protection of this data. My question is, can you store it or delete it or submit it to JICET or any NCBI or any other database like this?
Okay, so I think the Patrick database keeps all of the data private. Nothing is shared unless you want to make it public. But what the Vigor4 tool does is it gives you an output file that is very easy to upload to GenBank, if that's what you'd like to do. We don't have any direct connections with them yet, but that's something that we're exploring. But once you download your files, it should be very easy for you to submit them wherever you'd like in those formats.
Yeah, I know that's all easy. My question is, what is the policy of ViPR? Does ViPR protect this data? I mean, the data which I submitted, which is actually the data from NIH and from Pakistan. And I don't want it to be published in any other database. So do you keep it for some months or for some days, or just delete it? The FASTQs.
This is Richard. It will be maintained in your private workbench area, and you have the option of retaining it there or deleting it. And so if this is something that you're concerned about, what I would recommend is that you can run your sequences through the assembly and annotation service, get your result files, download those to your computer, and then delete them from your workbench. As Anna said, we do not release any of those sequences in the public part of the database. We never do that regardless. But to be extra, careful on your end, I would recommend just deleting them from your workbench when you're finished.
Yeah, that's very great. One last question. If we would like to collaborate with ViPR database, for example, from NIH, from National Institute of Health, Pakistan, Islamabad, so how could we do that? Can we do that? I mean, is there any possibility for collaboration with the ViPR database for analyzing those genomes?
Yes, we do collaborative projects with users based on requests like this. We've definitely collaborated with different external groups to do this kind of phylogenetic analysis and other kinds of comparative genomic studies, hopefully resulting in publications. And that is something that we do routinely do. And so I think the best option would be, as Anna mentioned, if you go to the ViPR website and on the home page, on the right-hand side, there are different options to contact us. If you do that, then we kind of keep track of these requests. But if you would like to establish a collaboration, that's the best mechanism to do that.
Yeah, thank you so much. That's great.
And we're happy to provide support.
Thank you so much. We would be happy to collaborate with you. Thank you.
Great.
Thank you. Thanks.
Would you mind just scrolling up to the top of the page and just showing everyone where the contact us link is? Under support on the top right.
Here.
And then you can just click on ask a question, and that will send you to a form where you can just send us any feedback or requests, or if you'd like us to provide any special training that's customized to your institute, or you have any other questions about collaborations. Okay, so if there are no other questions, thank you so much for attending, and we hope we see you at our next workshops.